stop sharing differences. Mull, red. Okay, ഞാൻ പോട്ടെ.
So welcome to all of you. So today's resource person, I welcome to today's resource person, like Vargeese sir. I welcome to you, sir.
Sir, welcome.
So and I welcome to our organization academy MD Managing Director Dr. A.B. Basaraju. I welcome to you, sir. And also I welcome to our CEO Dr. A.M. Ramesh sir. I welcome to you, sir. And also I welcome to all the today's attendees. And also I give a small introduction about Dr. P. Vargeese sir. Vargeese sir got an MSc in Botany, Mansa Gangoत्री, Mysore, and appointed as a PU College biology lecturer to Government PU College, Ilkal, Bagalkot district in 1993. Sir is presently working as a principal in Government PU College, Malpe, Udupi district. And sir completed 10 years service in Ilkal and transferred to Udupi Girls PU College in 2003 and continued as a lecturer up to 2018. And also he promoted as a principal in 2018 and having 25 years experience in lecturer series and experience in principal in 4 years. And also working as a resource person to PU College lecturers, and also he trained to high school teachers. So thank you. Over to you, sir.
Okay, thank you, sir. Hello, students. So let us discuss something about the chapter respiration in plants. And it's a very important thing. It's a very important chapter also. So one of the major things is that we need energy. Normally the energy comes from the food. You know, we eat the food digests in our gut, and then this digested food normally finally enter into the blood, and the blood supply everything to the cells. So for example, if you eat carbohydrate, glucose will be supplied to the cells. If you eat protein, amino acids will be supplied to the cells. So now, very important thing, what is the things happen that is after the digestion of carbohydrate, particularly starch. So once the starch is digested, it will convert it into glucose. So glucose will be supplied to all the cells because all cells require glucose. And the breaking of glucose in stepwise process release the energy. And normally we say, what is actually the respiration? Respiration is a method or a process during which energy releases from the food particles, especially carbohydrates. And we know very well, plants synthesize carbohydrate during which they utilize energy, that is sun energy. So sun energy is stored in the bond of glucose. When we break the glucose in our cells, the same energy will be released. But the sun energy always in the form of electromagnetic energy, whereas the energy what we release in the cells, that is in the form of chemical energy. So this is the difference. See, especially the sun energy store the plants in the form of chemical energy. And that chemical energy will enter into our body through the food. And once the food particles are broken, the same energy is released, the chemical energy is released in the form of ATP. And this ATP is utilized for all our activities. So there's a real thing of the respiration. Respiration actually an energy releasing process. Okay. So you're given all living organisms need energy for carry out daily life activities, be it absorption, transport, movement, reproduction, or even breathing. For everything, we need energy. So this chapter explains how could the energy released from the food particles. Again, a question arises. Is there any difference between the respiration in animals as well as in plants? Final process of respiration is similar in both plants and animals. But animals have certain certain system to make the process of respiration in a smooth way. Whereas no such systems are there in plants. Plants also respire, animals also respire. Respiration is the same. In both the cases, we we intake oxygen, and after breaking the carbohydrate molecules, carbon dioxide releases in every cell, and this carbon dioxide will be given out through the nose. The same thing happen in the plant. Then what it is given? Why it is given respiration in plants? So thing is very simple. In animals, breathing is there. Breathing, exchange of gases, then yeah, in external respiration, then transport of gases, then internal respiration. These are the few steps. They are there in animals. Those steps are not there in plant. The final steps in animals, that is cellular respiration. The only step in plant, that is also cellular respiration. So cellular respiration is common for both plants and animals. So this is a very important thing you should know. So when we ask some question to some students, what is there any difference between the respiration in plants and animals? Many students they give the answer that plants absorb carbon dioxide and give out oxygen. Animals absorb oxygen and give out carbon dioxide. So totally wrong. Respiration means there is only one definition. So that is taking oxygen in most of the cases. And after breaking the glucose, normally the carbon dioxide water are formed. Water will be there in body, and the carbon dioxide will be given out. These are final step are the only process that occur both in plants and animals. Here, there are some things to be studied here. Is it oxygen is essential for respiration both in plants and animals? It's not compulsory. More than 95% of the animals, 99% of the animals and plants, they utilize oxygen for respiration. And such a type of respiration, we call that as aerobic respiration. Okay. Whereas there are certain areas, certain reasons, or certain organisms, they respire in absence of oxygen. See, this respiration in absence of oxygen is called anaerobic respiration. So anaerobic respiration occur in some bacteria. Normally by lactic acid bacteria. So they are anaerobes. Not only lactic acid bacteria, there are many other bacteria also there, anaerobic bacteria. And anaerobic process also occur in our body cells, especially our muscle cells. That is skeletal muscle cells. So sometimes when we do some strenuous exercises, sufficient oxygen may not be supplied to the animal cells or our body cells, especially our muscle cells. In such cases, the cells shift from aerobic to anaerobic condition. So as a result, lactic acid will accumulate, and it is also a type of anaerobic respiration. So two types of respirations are there. One is aerobic respiration and another anaerobic respiration. One more very important thing. During aerobic respiration, the carbohydrates are completely oxidized. Once they oxidize completely, they release maximum energy. And that energy would be around 36 ATP. In old books, it is a 38 ATP. New books, about 36 ATP will be released. Whereas the same glucose molecule, if it is respired or it is oxidized in absence of oxygen, complete oxygen will not occur. Incomplete oxidation occur. Therefore, that releases only 2 ATP molecules. Very little amount of energy will be released when the same glucose molecule is broken without oxygen. With oxygen, without oxygen, there's a major difference. So maximum energy will be released from during the aerobic respiration, and very little amount of energy will be released during anaerobic respiration. Okay. So here there are also a few statements are there. Even in green plants, all other organs, tissues and cells, the non-green need food for oxidation. Hence, food has to be transferred to all non-green parts. Animals are heterotrophic. They obtain food from plants or indirectly. Saprophytes like fungi are dependent on dead and decaying animal. These are importance of food. Other things are given. See, what is important to recognize that ultimately all the food that is respired for live processes come from photosynthesis. I already explained. This chapter deals with the cellular respiration, the breakdown of food molecules or materials within a cell to release energy. Listen, respiration means it is a process to release energy, the energy already accumulated in the bonds of food molecules, and a trapping for this energy for the synthesis of ATP. So ATP is an energy currency. So we can utilize at any time. So that's why it will be there in the form of ATP. Okay. See, these all are the introductory part. Much more importance is not there to these things. See, I said already ATP acts as an energy currency of the cell. This energy trapped in ATP is utilized in various energy requiring processes of organism. And the carbon skeleton produced during the respiration is used as a precursor for biosynthesis of other molecules. See, the question again, I asked already, do plants breathe? Yes. Well, the answer is to this question is sorry. Answer to this question is not quite so direct. Yes, that is the answer. Plants require oxygen for respiration to occur, and they also give out carbon dioxide. Hence, plants have systems in that ensure the availability of oxygen. The question arises: how can the plants get oxygen? There are methods are there. Number one, during photosynthesis, oxygen will be released one of the byproduct. This oxygen also can be used by the plant cells during the respiration. Otherwise, sometimes they get oxygen from the atmosphere. But oxygen from the atmosphere utilization is very less. Most of the cases, the plants utilize the oxygen released during the photosynthesis. Okay. Here also given, plants, unlike animals, have no special organ for gaseous exchange, but there's stomata and lenticels for this purpose. There are several reasons why the plants get along. There are several reasons why plants can get along without respiratory organs also. So there are two reasons are there. It's an important one. Each plant part takes care of its own gas exchange needs. So there is very little transport of gases from one plant to another part. Transport of gas is very rare. Second one, plants do not present great demand for exchange of gas exchange. Root, stem, and leaves respire at rates far lower than animals do. Only during photosynthesis are large volume of gases exchange, and each leaf is well adapted to take care of its own needs during these periods. Okay. When cells photosynthesize, availability of oxygen is not a problem in this case, in the C cells, since oxygen is released within the cell. This is one of the things. Means whatever the cells, green cells are there in the plants, they photosynthesize. And during photosynthesis, oxygen will release naturally as a byproduct. This oxygen will be used by the same cells for their respiration. That's one of the things. Second point. Third one, the distance that gases must diffuse even in large, bulky plants is not great. Each living cell in a plant is located quite close to the surface of the plant. This is true for leaves. You may ask, but what about the trunk, wood, stem, and other things? It's a very simple question. Living tissue in a plant, especially in woody plants, present on the peripheral region, not in the inner surface or inner part of the plant body. Inner part of the plant body is made up of wood or xylem. So xylem is a dead tissue. They no need of any oxygen. Oxygen required to the living part of the plant. So those cells will be there in the peripheral region. Therefore, they make use of the oxygen directly from outside through the lenticels or through stomata and other things. Okay. So that is important thing. Again, we want to know what the equation for the respiration. Okay. So here, respiratory equation C6H12O6 plus 6O2. It gives 6 carbon dioxide. Then H6H2O and energy. So this is the equation. Okay. If you want to know very clearly, see when you reverse this same thing, it will be the reaction of a photosynthesis. Carbon dioxide, water plus energy. It gives glucose and oxygen. Meaning is that photosynthesis and respiration is a quite opposite phenomenon. During photosynthesis, sunlight energy will be utilized, whereas during respiration, the same energy is released in the form of chemical energy, that makes the ATP. So this is a very important one. And here what I explained here, how to that breaking of glucose that occur in each and every cell. So we supply glucose to each and every cell of our body, especially through the medium of blood. Okay. See, and this glucose from the blood, I mean, I said it is supplied to the cells. And same way, normally in human body, we respire. As a result, oxygen will enter into our blood. This blood also we supply to the cells. Same thing happen in plant, but in different method, in a different way. I said glucose will be synthesized by the plant itself, and the glucose will be supplied to all non-green part of the plant body, and oxygen will be released in green cells of the plant body during photosynthesis, whereas non-green part of the plant body get oxygen from either the stomata or through the lenticels. Thereby they fix or they find out the source for oxygen. Okay. Again, another important thing, I said already, during the process of respiration, oxygen is utilized and carbon dioxide, water and energy is released as a product. Okay. So here, yeah, so all these things I think I explained. The term, there is one of the very important thing. What are the events occur during the respiration? During cellular respiration, I said there are two types of respirations are there. One is aerobic respiration, another is anaerobic respiration. Aerobic respiration takes place in presence of oxygen. Anaerobic respiration takes place in absence of oxygen. During aerobic and anaerobic respiration, some steps occur because respiration is not a single step process. It's a multi-step process. During aerobic respiration, the first step is the glycolysis. Second one, Krebs cycle. Third one, ETS, electron transport system. These three events occur during aerobic respiration. What about during anaerobic respiration? Here, two steps is there. Again, anaerobic respiration are two types are there once again. Number one, alcoholic fermentation. Another one is lactic acid fermentation. During alcoholic fermentation, glycolysis is the first step. Then second one, formation of ethyl alcohol and carbon dioxide. Two steps. During lactic acid fermentation, again here also two steps are there. Glycolysis is the first step. Second step, that is what you call formation of lactic acid. Okay, formation of lactic acid. Listen carefully. In all these three things, what I said about aerobic respiration, alcoholic fermentation, lactic acid fermentation, in all these three processes, glycolysis is a common pathway. Glycolysis is a common pathway. The question arises, what are the events that occur during glycolysis? Before that, I want to explain a few things about the glycolysis. Glycolysis is an anaerobic process. No need of oxygen. Anaerobic occur in both, glycolysis occur both in anaerobes and aerobes. It is an anaerobic process. It occurs both in aerobes and anaerobes. And the end product of glycolysis, in any cases, that is pyruvic acid. Two molecules of pyruvic acid. Again. For glycolysis to occur, no need of any cell organelles. It occurs in the cytoplasm. So cytoplasm will be there in both aerobes and anaerobes. Okay. So these are the very important things to be known. And glycolysis was traced by three people. Their name, Emden, Mayor of Parnas. Emden, Mayor of Parnas. Emden, capital E, Mayor of capital M, Parnas, capital P. the first letters of their name. Therefore, this is shortly called EMP pathway. So EMP pathway, glycolysis. Okay. So this is an anaerobic process. No need of any cell organelles for this. End products are pyruvic acid, 2 molecules of pyruvic acid, and no carboxylation occur, no oxidation occur. Okay. But it releases about 2 ATP molecules and 2 NADPH2 molecules. Okay, so this is a very important things about glycolysis. So glycolysis is a 10-step reaction. 10-step reaction. Okay, so glycolysis, the starting one is glucose. If a starch is there in the stored cells, starch will finally convert it into glucose. So glucose is the starting point for glycolysis. Actually, what do you mean glycolysis? Breaking down of glucose into pyruvic acid by, various steps of enzymatic reactions. Okay, and the release of about 2 ATP molecules and 2 NADPH2 molecules. This is a clear things about glycolysis. Okay, yeah. So here everything is given. All these things what I explained is there in the explained. What are the things I explained so far? All are there in the notes. Okay. See here, M done, Gasto M done, Autograph, and J Parnas. So simply called EMP path. Okay. See, these are cycle of glycolysis. So 10-step reaction, glucose. See, here the two steps, two steps are there again in glycolysis. First one, the energy utilizing steps, whereas second one, energy gaining process or energy releasing process. So the first steps in the first half of the glycolysis, energy consuming process is there. See, listen, glucose converted into glucose 6-phosphate, 1 ATP is utilized. Enzyme that is hexokinase. Then glucose 6-phosphate will convert to fructose 6-phosphate. That is by a process called isomerization. Enzyme isomerase will be there. And one more ATP is utilized by this fructose 6-phosphate to become fructose 1,6-diphosphate. Okay, so 2 ATP molecules are utilized, means loss. Then fructose 1,6-diphosphate will changes to become that is phosphoglyceraldehyde. Glyceraldehyde phosphate. So this glyceraldehyde phosphate, which undergo that means a 6C compound. Glucose, fructose, everything. They are 6C compound. The fructose 1,6-6C compound that cleaved to become phosphoglyceraldehyde and DHAP. So DHAP, phosphoglyceraldehyde, both are 3C compound. Fructose 1,6-diphosphate cleaved. So cleaved means 6C compound will changes to become 3 compound of 2 molecules. Final, this phosphoglyceraldehyde, we should consider 2 phosphoglyceraldehyde molecules are there because DHAP and phosphoglyceraldehyde both are interconvertible compounds. So there are 2 are there now. One phosphoglyceraldehyde on DHAP, dihydroxyacetone phosphate. So both are interconvertible. Therefore, you should consider as 2 phosphoglyceraldehyde or 2 DHAP. So you should consider all are in 2 molecules. Okay. Again, the phosphoglyceraldehyde, 2 molecules of phosphoglyceraldehyde, they undergo oxidation. Oxidation here means not addition of oxygen, removal of hydrogen. So that hydrogen will be taken by 2 molecules of NAD, and 2 molecules of NAD becomes 2 molecules of NADH. Sorry, NADP. It is not NADPH, it is NADH. Okay. NADPH comes in the photosynthesis. NADH comes that is in the respiration. So NADH. And as a result of this, these 2 molecules of phosphoglyceraldehyde changes to become 2 molecules of 1,3-bis-phosphoglyceric acid or glycerate. Then once again, 2 ADP molecules will convert to ATP molecules because energy is released. That energy is used for the synthesis of ADP to ATP. Okay. As a result, the 2 molecules of ATPs are synthesized. Clear? And these 2 molecules of ATPs are there now. They, as a result of that, Bisphosphoglycerate is changed to become 3-phosphoglycerate because the phosphate from first position will be released. That P will added to the ADP, so ADP becomes ATP. Because the two molecules are there, two phosphates are released, 2 ADP becomes 2 ATP. And the final the product will be 3-phosphoglycerate. So this 3-phosphoglycerate again undergo a changes. What do you mean? What is the changes occur here? The phosphate will be shipped from second carbon to third carbon. Sorry, third carbon to second carbon. As a result, 3-phosphoglycerate will changes to become 2-phosphoglycerate. Okay. Again, this 2-phosphoglycerate is there now. It loses, it gets a molecule of water to possum. Sorry, loses a molecule to param phosphoenol pyruvate. Param phosphoenol pyruvate. Okay. So this phosphoenol pyruvate again loses phosphate molecules. Energy also released. That energy is used for the synthesis of ATP molecule. And finally, 2 molecules of pyruvic acids are formed. So this is the end product of glycolysis. Okay. Here, what are the things to be known? First of all, you know, I said already the characteristics of glycolysis. Glycolysis occurs in 10 steps. Each and every step is a separate enzymes. They participate in the in the reactions or in the processes. Separate enzymes are there. Okay. And final product of 2 molecules of pyruvic acid. Here, one thing very importantly you should be, consider. You know what is that? Glucose converted into pyruvic acids. You know the number of carbon in the glucose. Number of carbon in the glucose. 6 carbon. What is the number of carbon in the pyruvic acid? 3. So 3 carbon compound. Such 2 molecules are formed. So 2 molecules of 3 carbon means 6 carbon. What it means now? No carbon dioxide is released or no carbon dioxide is taken in. Okay. That's a very important thing. And again, I said the preparatory step, 2 ATP molecules are utilized. And again, in the gaining stage, last stages, 4 ATP molecules are formed. When 4 ATP molecules are formed, 2 already utilized, no? So you should take out 2. 4 ಎರಡು ಮಾಲಿಕ್ಯೂಲ್ ಯೂಸ್ ಆಗಿದೆ, 4 ಮಾಲಿಕ್ಯೂಲ್ಸ್ ನಾವು ತಗೊಂಡಿದ್ದೇವೆ ಅಂದ್ರೆ ಬಂದಿದೆ. ಸೊ ಆ 4ರಲ್ಲಿ 2 ಕಳೆದ್ರೆ 2 ATP molecules will gain and 2 NADH2 molecules also gain. 1 NADH2 molecule is equal to 3 ATP. ಅಂದ್ರೆ ನಾರ್ಮಲಿ 3 ATP ಅಂದ್ರೆ 2 NADH2 molecules ಅಂದ್ರೆ 6 ATP plus 2 ATP as ATP itself. So totally 8 ATP is a total gain. ಆಯ್ತಾ? ಸೊ ಆದ್ರೆ ಕೆಲವು ಬುಕ್ 6 ATP ಅಂತ ಕರೀತಾರೆ. Don't worry. So it is totally 8 ATP ಇಲ್ಲಿ ಬರುತ್ತೆ. Clear? When here 8 ATP you consider, total ATP formed during the aerobic respiration will be 38. If you consider only 6 here, total ATP will be 36. But that 2 ATP difference ಏನು ಸರ್ ಅಂದ್ರೆ the pyruvic acid what are formed here, no, it should be shifted from the cytoplasm into mitochondria. For that they utilize 2 ATP molecules. Again to ಬಂದ್ರೆ R ಅಂತಾನೆ consider ಮಾಡೋದು because 2 again it is utilized. Okay. So this is glycolysis. This entire thing that what I explained so far. And each and every step in enzyme I explained ಮಾಡಿದ್ದಾರೆ. So here also enzyme you can see. I mentioned enzyme also here also. I mean in this schematic representation also. Phosphoglucoisomerase, phosphofructokinase, aldolase, a cleaving enzyme. Then phosphoglyceraldehyde hydrogenase, phosphoglycerate kinase, phosphoglycerate mutase, enolase, pyruvate kinase. These are the enzymes. The same steps are explained in your entire part of these few chapters. Okay. So ATP is utilized in 2 steps. First step, conversion of glucose into glucose 6-phosphate. 1 ATP is utilized. So the same thing I explained so far. It is given in entire these pages. No need to worry about this. Okay. So they say where are the things, where are the reasons where the ATPs are synthesized. Clear? See, if it is anaerobic respiration, it occurs in some prokaryotes and also eukaryotes. Prokaryotes especially, that is in lactic acid bacteria. And some fungus also. Sorry, fungus especially, that is eukaryote. Yeast is another organism. They respire anaerobically. Okay. One more thing. I said already that muscle cells of higher animals, skeletal muscle. But skeletal muscle is a eukaryotic cell. What I'm saying is anaerobic respiration occurs both prokaryotes and eukaryotes. Okay. What about aerobic respiration? Aerobic respiration also occur in both prokaryotes and eukaryotes. But what about in bacteria? There are some aerobic bacteria are there, some anaerobic bacteria also there. Aerobic also there, anaerobic also there. Therefore, respiration, especially aerobic and anaerobic, both occur both in prokaryote, eukaryote. Okay. Both prokaryotes and eukaryotes. That's the meaning. So there is no discrimination about prokaryote and eukaryotes. Especially for this things. Clear? Sometimes you may ask a question that in prokaryotes, normally in eukaryotes, that cellular respiration occur, aerobic respiration occur in a mitochondria. Is it there any mitochondria in a prokaryotes? No. But in prokaryotes, there are mesosomes, especially in bacteria. In mesosomes, there are oxysomes are there. The mesosomes and oxysomes together perform the function of mitochondria. Okay. So now come to the anaerobic process. Already I said in anaerobic also, or anaerobic otherwise fermentation. Okay. In fermentation also, this is there. What is called glycolysis there. What happened after glycolysis in anaerobic organisms? Very important one. In fermentation, say by yeast, in incomplete oxidation of glucose is achieved under anaerobic condition by set of reactions where pyruvic acid converted into carbon dioxide and ethyl alcohol, means ethanol. It is alcoholic fermentation. Okay. The enzyme pyruvic acid decarboxylase and alcohol catalyzes this reaction. So in other organisms like some bacteria, they produce lactic acid from the pyruvic acid. When lactic acids are produced, there is no production of carbon dioxide. So carbon dioxide production occurs only during during the alcoholic fermentation, not during lactic acid fermentation. Okay. So here also the test steps is like that. In animal cells, I said in our muscles, during strenuous exercise, oxygen supply will be very less, inadequate oxygen supply. So in such cases, these muscle cells, they shift from aerobic to anaerobic, and during which pyruvic acid molecules are directly changed to lactic acid. That enzyme is lactate dehydrogenase. Okay. So during which the NADH2 molecules are utilized, which are synthesized during the glycolysis. During alcoholic fermentation also, the NADH2 molecules which are synthesized during the or produced during the glycolysis will be utilized. Therefore, the NADH2 molecules, what I said in the few minutes before, that a NADH2 molecule can give rise to 3 ATP molecules. I said no. So when lactic acid fermentation or alcoholic fermentation occur, these NADH2 molecules are utilized. Therefore, there is no generation of ATP from NADH2. Only the 2 ATP remains now during glycolysis. That will be the end product energy or end energy or the energy produced at the end of the fermentation, means alcoholic fermentation or lactic acid fermentation. Okay. So that's the thing. Here is there now glucose, glyceraldehyde phosphate. So that is what you call pyruvic acid. Here now, this step is the glycolysis. Up to here, this is glycolysis, and then lactic acid. When lactic acid occur, NADH2s are utilized. Ethyl alcohol carbon dioxide also, the NADH2s are utilized. This is a schematic representation. See, that is the entire thing is there. I already explained. Come to this one, aerobic respiration. Here also I said that glycolysis is the first step, and that 2 molecules of pyruvic acid are formed. Okay, and 2 molecules of NADH2 are formed. 2 molecules ATP also formed. But this NADH2 and ATP what are formed during the aerobic process of glycolysis, they are not reutilized. They will stay as such, and that will form as in the form of energy. Okay, see, The complete oxidation of pyruvic acid by stepwise removal of all the hydrogen atoms, leaving 3 molecules of carbon dioxide. The passing on the electron removed as part of hydrogen atoms to molecule of oxygen with the simultaneous sense of ATP. ಇದಾಗ್ತದೆ ಇಲ್ಲಿ ನೋಡಿ. This is a very important step. What happened to the pyruvic acid during anaerobic respiration? Pyruvic acid combines the coenzyme A, and in presence of NAD, with the help of NAD, the Mg2 plus ions, metal ions are essential. Pyruvate dehydrogenase enzyme also required. As a result, two things happen. One is oxidation, another decarboxylation. You know what I mean oxidation? Here, removal of hydrogen from the pyruvic acid. That hydrogen will be taken by NAD. NAD become NADH or NADH2. That's why written in the form of NADH plus H plus, or you can say NADH2. Okay, that is as a result of oxidation. What about the other process, carboxylation? As a result, a carbon dioxide molecule is released. You know, I said in a pyruvic acid there are 3 carbon molecules are there, of which 1 carbon will be removed. So that carbon dioxide will be released out. As a result, the 3 carbon compound pyruvic acid changes to 2 carbon compound. That 2 carbon compound is the see this acetyl CoA. Okay, so there is a 2 carbon compound. Acetyl CoA is a 2 carbon, whereas 2C compound, whereas pyruvic acid is a 3C compound. Okay, so what happened 1C? This the C is the here carbon dioxide. So as a result, 3 carbon will changes to become 2 carbon. Okay, see, this is a very important step. The question appeared many times in the NEET examination. That is, what happened to the pyruvic acid during aerobic respiration? Number 1, oxidation. Second, decarboxylation. Okay. Then, what happened to after that? Acetyl CoA. So the acetyl CoA enter into the mitochondrial matrix. You know, mitochondria is a 2 membrane, outer and inner membrane. This passes through the both the membranes and finally they reaches the matrix. In matrix, already there is a compound in the matrix. Oxaloacetic acid. See, by this step I can explain everything. Oxaloacetic acid. Pyruvic acid is a preparatory step. Pyruvic acid converted to acetyl CoA. So acetyl CoA enters into matrix, where in the matrix there is oxaloacetic acid, OAA, and this is a 2C compound, is a 4C compound. This 4C plus 2C together form a 6C compound, that is citric acid. In presence of an enzyme, citrate kinase, some enzymes will be there. So in presence of enzyme, it will convert into citric acid. So citric acid again undergo 2 processes, decarboxylation and oxidation. As a result, it becomes alpha ketoglutarate acid. 6 carbon changes to 5 carbon because 1 carbon is removed here. Then alpha ketoglutarate acid will changes to become succinic acid. Here many things happen. Decarboxylation occur, oxidation occur, phosphorylation occur. So you should know what are the events occur during the conversion of each and every steps. Decarboxylation, removal of carbon dioxide, oxidation, H plus is removed, which is accepted by NAD. NAD become NADH2, and the GDP will be changes to become GTP. So GTP is equal to AATP. 1 GTP is equal to ATP. After all these process, the alpha ketoglutarate acid changes to become succinic acid. So succinic acid again undergo oxidation, but at this time, this hydrogen is not taken by NAD, but taken by FAD. Okay, FAD becomes FADH2. So succinic acid changes to become malic acid. So here, if you go to some other reference books, you will get a few more steps here. Intermediate steps are there. Fumaric acid also formed here. But in your book, it is given only this step. Don't go for that. You use this one only. So this malic acid is a 4C compound because no carbon dioxide changes occur. Malic acid once again undergo oxidation. As a result, the NAD will be taken the H plus to become NADH. Malic acid changes to become oxaloacetic acid, and it is ready to accept another pyruvic acid molecule going to enter into the matrix of mitochondria through the membrane after that oxidation and decarbonation to form acetyl CoA. No, this process will cycle. This cycle will continue. Here, what you should know? No, see here there is an NADH2 is formed. One NADH2 means 3 ATP. In another NADH2, again 3 ATP is also formed here. Here there is another NADH2. It makes 3 ATP. Okay. So here again there is one more NADH here NADH2. Here also there are 3 ATP. Okay. See how many ATPs are formed here? 3 plus 3. 3 plus 3 plus 3. 3 plus 3 6. 6 plus 3 9. 9 plus 3 12. So 12 ATP from NADH2. And this FADH2 is there. No, it is able to synthesize 2 ATP. 2 ATP. So that is 2 more ATP. And here this GTP is there. GTP. 1 GTP means 1 ATP. 1 GTP means 1 ATP. Totally 15 ATP from one molecule of pyruvic acid. So I said there are 2 molecules of pyruvic acid are formed for glucose. Is it or not? So 15 more ATPs are formed. So 15 plus 50, totally 30 ATP molecules are formed from this one. And I said already 8 ATP molecules are synthesized during from glycolysis. That 30 ATP, this is 30 plus 38, totally become 38 ATP. 2 ATPs are used for the carrying of pyruvic acid from cytoplasm into mitochondria. So you should less 2 from 38, it will be 36. But for some books explain it as a 38, some books explain it as a 36. Don't worry, one will be given in your question paper. So you should find out whether there is a 36 or 38. So sorry, one will be there, not both 36 and 38. One will be there. You should consider the 36. Okay, so that is the Krebs cycle. And the another important thing about this Krebs cycle is that another important thing. All these things I explained in the about this one. Another is ETS. This is a very, very important thing. What is the ETS? These are sole things to be studied. I just try to explain within 10 minutes of time. We no need of 10 minutes. Listen. See, this is the electron transport system. It occurs on the inner surface of inner membrane of mitochondria. And there are electron transport system also there, proton transport system also there. See, this yellow color is there. No, it is a proton transport one. Whereas this yellow color itself is the electron transport also. It transports electrons and proton. But thing is that this FMN is there now. It transport proton from, this is the matrix side. Okay, this is a matrix side. So in matrix side, the NADH2 is formed during Krebs cycle. So NADH2 supply 3 pairs of protons through this protein channel. So 3 pairs. Okay, each pair of proton means H plus, is able to synthesize 1 ATP molecule. One pair of proton is able to synthesize 1 ATP molecule. That much of energy will be there in that one. So when 3 pairs they supply, that will be 3 ATP molecules synthesized by this one. So I said no, that NADH2 will produce 3 ATP. So that is simply theoretically explained. This is a practical process. This is the stage where that NADH2 are converted to ATP. Normally, while we explain the Krebs cycle, we say that 3 ATPs are synthesized from 1 NADH2. It's a simply theoretical one. This is a practical one. Okay, see here. This NADH2 synthesized in the Krebs cycle supplies 3 protons through this one. The proton will be given to this place. It's the outer space. It's the inner space. This is the inner membrane. Somewhere here it is the outer membrane. This space is the inner space. Sorry, it's the outer space. This one is the inner space. From inner space to outer space, 3 pairs of protons are pumped through the protein FMN. While it also supplies electron. Electron will not go out to outer space. Electron will not go to outer space. Electron will travel in this method. See this on the inner surface of the inner membrane. Okay. So this is the track of electron transport. So electron from this FMN come to ubiquinone. Ubiquinone, meanwhile, ubiquinone also able to, ubiquinone are again proton and electron carriers. Okay, they also supply electron from they also supply proton from inner surface to outer surface through ubiquinone. It is a mobile proton carrier. It's also electron carrier, but it electron will be supplied to the next stage, that is cytochrome BF and other things are no. So they supply the electron to cytochrome B. Cytochrome B further supply the electron to ubiquinone. Okay, again here also ubiquinone is a mobile electron and proton carrier. Whereas this cytochrome B is there, no, it is only electron carrier, not proton carrier. Again here there is a Racker particle is there. I will explain about this one later. So this electron finally come to again another electron carrier, that is cytochrome cytochrome A1 and cytochrome A1, cytochrome A3. See, so this one is also a electron carrier. So this electron, what is pushed here, it will travel through this membrane. Finally, they return back into the matrix through this F0 F1 particles. This F1 by F0 F1 particle, it is nothing but okay. So this one, electrons are travel only on the inner surface of membrane, whereas proton they supply. See, listen, here this ubiquinone is a no. This ubiquinone supply hydrogen that is donated by FADH2. FADH2 is able to push two pairs of protons into the outer space. As a result, each pair of proton is able to synthesize 1 ATP, I said no. So FADH2 is able to produce 2 ATP. Okay, that does pushes the proton through ubiquinone. There any other proton carriers or proton are there in the matrix will be supplied to this ubiquinone. In such a way, the proton supplied to outer space, whereas electron travel on the inner surface of the membrane, finally that return back to matrix. See, when proton accumulate in the outer membrane, there is a proton motive force will be formed, means accumulated or developed. This proton motive force is able to push two protons at a time into the matrix through this F0 F1 particle. Okay, see, through this F0 F1 particle, two protons each will be supplied from outer space to inner space. ಇಲ್ಲಿರ್ತಕ್ಕಂಥ ಪ್ರೋಟಾನ್ಸ್ ಏನಿದೆಯಲ್ಲ ಇವುಗಳಿಗೆ ಪ್ರೆಷರ್ ಜಾಸ್ತಿ ಆಗುತ್ತೆ, ಅವಾಗ ಇಟ್ ಫಾರ್ಮ್ಸ್ ಪ್ರೋಟಾನ್ ಮೋಟಿವ್ ಫೋರ್ಸ್. ಈ ಪ್ರೋಟಾನ್ ಮೋಟಿವ್ ಫೋರ್ಸ್ ತಕೊಂಡು ದಟ್ ವಿಲ್ enter into the matrix through this F0 F1 particle. F0 particle is there in the membrane, F1 particle will be there in the matrix. It's a protrude into the matrix. ಅದು ಕರೆಕ್ಟ್ ಆಗಿ ಗೊತ್ತಿರಬೇಕು. ಈ ಪೋಟಾಮೋಟಿವ್ ಫೋರ್ಸ್ ಇಂದ ಪ್ರೋಟಾನ್ಗಳು they pass, mean they move through this one, reaches here. This while the proton move, ಇಲ್ಲಿ ಅದು ATP ಫಾರ್ಮೇಶನ್ ಆಗೋದು F1 particle ಅಲ್ಲಿ, that membrane permit to be changed and ADP will convert into ATP. For that it requires energy. That energy it will be given from the protons. ಆ proton ಅಲ್ಲಿ ಎನರ್ಜೆಟಿಕ್ ಇರುತ್ತೆ, ತುಂಬಾ ಎನರ್ಜಿ ಇರುತ್ತೆ. That energy will be utilized for the synthesis of ATP from ADP. ಓಕೆ, ಅಂದ್ರೆ ಎರಡು ಪ್ರೋಟಾನ್ ಬರುವಾಗ ಸಾಕಷ್ಟು ಎನರ್ಜಿ ಜೊತೆಗೆ ಇರ್ತದೆ. ಆ ಎನರ್ಜಿನ ಯೂಸ್ ಮಾಡಿ ADPಗಳು this proton will release into the matrix. ಈ ಮ್ಯಾಟ್ರಿಕ್ಸ್ ಅಲ್ಲಿ ಬಂದ ಪ್ರೋಟಾನ್ ಇಲ್ಲ, ದೇ ಆರ್ ಅಗೇನ್ ಹೈಲಿ ಎನರ್ಜೆಟಿಕ್. ಇದಕ್ಕೆ ಸಾಕಷ್ಟು ಎನರ್ಜೆಟಿಕ್ ಇರೋದು ಇದು. ಓಕೆ, ಈ ರೀತಿ ಎನರ್ಜೆಟಿಕ್ ಇರೋದ ಪ್ರೋಟಾನ್ ಅನ್ನ ನಲ್ಲಿಫೈ ಮಾಡ್ಬೇಕಾದ್ರೆ ವಿ ನೀಡ್ ಆಕ್ಸಿಜನ್. ಈ ಎಲೆಕ್ಟ್ರಾನ್ ಏನ್ ರಿಲೀಸ್ ಟ್ರಾವೆಲ್ ಆಗಿ ಬಂತಲ್ಲ, ದಿಸ್ ಆಕ್ಸಿಜನ್ ವಿಲ್ ಬಿ ಟೇಕನ್ ಬೈ ಹೈಡ್ರೋಜನ್ and hydrogen gets a charge, a negative charge, and this is the proton. Two protons and one hydrogen form a water. ಇದನ್ನ metabolic water ಅಂತ ಕರೀತಾರೆ. H2O will be formed. ಅಂದ್ರೆ ನೀವು ಉಸಿರಾಡಿದ್ರಲ್ಲ, ಆಕ್ಸಿಜನ್ ತಗೊಂಡ್ರಲ್ಲ, ನೀವಾಗ್ಲಿ ಪ್ಲಾಂಟ್ಸ್ ಆಗ್ಲಿ, ಆಕ್ಸಿಜನ್ ಎಲ್ಲಿ ಯೂಸ್ ಆಯ್ತು? ಕ್ರೆಬ್ ಸೈಕಲ್ ಯೂಸ್ ಆಗ್ಲಿಲ್ಲ. ಆಲ್ರೆಡಿ ಐ ಸೆಡ್ ಯಾವ್ದು ನಿಮ್ಮ ಗ್ಲೈಕೋಲೈಸಿಸ್ ಆಲ್ಸೋ ಎನಾರೋಬಿಕ್. ಹೌದಾ? ಕ್ರೆಬ್ ಸೈಕಲ್ ಯೂಸ್ ಆಗ್ಲಿಲ್ಲ. ಇದು ಕ್ರೆಬ್ ಸೈಕಲ್ ನ ಭಾಗ. ETS ಬೇರೆ ಅಲ್ಲ, ಕ್ರೆಬ್ ಸೈಕಲ್ ಪಾರ್ಟ್ ಆಫ್ ಕ್ರೆಬ್ ಸೈಕಲ್. ಹಾಗಾಗಿ ಕ್ರೆಬ್ ಸೈಕಲ್ ನ ಫಸ್ಟ್ ಪಾರ್ಟ್ ಯೂಸ್ ಆಗಲಿಲ್ಲ. ಸೆಕೆಂಡ್ ಪಾರ್ಟ್ ಏನಿದೆಲ್ಲ ETS, ಎಲೆಕ್ಟ್ರಾನ್ ಟ್ರಾನ್ಸ್ಪೋರ್ಟ್ ಸಿಸ್ಟಮ್, ಆಲ್ಸೋ ಟರ್ಮಿನಲ್ ಆಕ್ಸಿಡೇಷನ್, ಅಲ್ಲಿ ಇರ್ತಕ್ಕಂಥ ಪ್ರೋಟಾನ್ ಅನ್ನ ನಲ್ಲಿಫೈ ಮಾಡ್ಲಿಕ್ಕೆ ಮಾತ್ರ ನೀವು ಆಕ್ಸಿಜನ್ ಯೂಸ್ ಮಾಡೋದು. ನಾರ್ಮಲಿ ವಿ ಎಕ್ಸ್ಪ್ಲೈನ್, ವಿ ಬ್ರೇಕ್ ದ ಗ್ಲೂಕೋಸ್ ಬೈ ಯೂಸಿಂಗ್ ಆಕ್ಸಿಜನ್ ಅಂತ. ಆಕ್ಸಿಜನ್ ತಗೊಂಡು ಯಾವ ಗ್ಲೂಕೋಸ್ ಅನ್ನು ಕಟ್ ಮಾಡಲ್ಲ. ಆಕ್ಸಿಜನ್ ಇರೋದು ಯಾತಕ್ಕೆ ಅಂದ್ರೆ ಫೈನಲ್ ಆಗಿ ಬರ್ತಕ್ಕಂಥ ಈ ಪ್ರೋಟಾನ್ ಗಳನ್ನ ನಲ್ಲಿಫೈ ಮಾಡ್ಲಿಕ್ಕೆ. ಇಲ್ಲ ಅಂದ್ರೆ ವಿತ್ ಇನ್ ನೋ ಟೈಮ್ ದ ಪರ್ಸನ್ ವಿಲ್ ಡೈ. ಆ ಪ್ರೋಟಾನ್ಗಳು ಅಷ್ಟು ಎನರ್ಜಿಟಿಕ್, ಅದು ಎಲ್ಲದನ್ನು ನಾಶ ಮಾಡಿಬಿಡ್ತದೆ. ಆಕ್ಸಿಜನ್ ನಲ್ಲಿಫೈ ಮಾಡ್ಲಿಕ್ಕೋಸ್ಕರ ಮಾತ್ರ ನಾವು ಆಕ್ಸಿಜನ್ ತಗೊಳ್ಳೋದು. ಒಂದ್ ವೇಳೆ ನಿಮ್ಮ ಕುತ್ತಿಗೆಯಾರ ಹಿಡ್ಕೊಂಡ್ರೆ ನಿಮಗೆ ಆಕ್ಸಿಜನ್ ಸಿಗ್ಲಿಲ್ಲ ಅಂದ್ರೆ, ಇಲ್ಲಿ ಆಕ್ಸಿಜನ್ ಸಪ್ಲೈ ಆಗಲಿ ಅಂದ್ರೆ ಈ ಪ್ರೋಟಾನ್ ನಿಮ್ಮನ್ನ ಸಾಯಿಸೋದು. ಆಕ್ಚುಲಿ ನಿಮ್ಮ ಕುತ್ತಿಗೆ ಯಾರು ಬಿಗಿ ಹಿಡ್ಕೊಂಡು ಸಾಯೋದಿಲ್ಲ, ನೇಣಾಕೊಂಡ್ರೆ ಸಾಯೋದಿಲ್ಲ. ಆಯ್ತಾ? ಸಾಯೋದು ಯಾವ್ದಂದ್ರೆ when anything such things happen, you will not get sufficient oxygen to your cells. ಸೆಲ್ಲಿಗೆ ಆಕ್ಸಿಜನ್ ಬರಲಿಲ್ಲ ಅಂದ್ರೆ ಈ ಪ್ರೋಟಾನ್ ಕಂಟ್ರೋಲ್ ಮಾಡಕ್ಕಾಗಲ್ಲ, ಆ ಪ್ರೋಟಾನ್ ನಿಮ್ಮನ್ನು ಸಾಯಿಸೋದು. ಸೊ ದಟ್ ಈಸ್ ದ ಡೆತ್ ವೆನ್ a person, I mean, if he not get sufficient oxygen. ಅರ್ಥ ಆಯ್ತಾ? ಉಸಿರುಗಟ್ಟು ಸತ್ಯವಂತೆ, ಉಸಿರುಗಟ್ಟದ ಸಾಯೋದು ಯಾಕೆ? ಬಿಕಾಸ್ ಆಫ್ ಪ್ರೋಟಾನ್ಸ್. ಆ ಪ್ರೋಟಾನ್ಗಳು ರಿಲೀಸ್ ಆಗೋದು. ಸೊ ದಿಸ್ ಇಸ್ ದ ಇಂಪಾರ್ಟೆಂಟ್ ಥಿಂಗ್ ಒನ್ ಶುಡ್ ನೋ ಅಬೌಟ್ ETS. ಆಯ್ತಾ ಮಕ್ಕಳೇ? ನೋಡಿದ ಪೂರ್ತಿ ಸ್ಟ್ರಕ್ಚರ್ ಕಂಪ್ಲೀಟ್ ಆಗುಂಟು. ಸರಿಯಾಗಿ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. ವಾಟ್ ಐ ಎಕ್ಸ್ಪ್ಲೇನ್ ಸೋ ಫಾರ್, ದೇರ್ ಆರ್ ಇದುಂಟು ಕಾಂಪ್ಲೆಕ್ಸಸ್ ಉಂಟು, ಅದನ್ನ ಚೂರು ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. ಕಾಂಪ್ಲೆಕ್ಸ್ ಆಲ್ಸೋ ವೆರಿ ಇಂಪಾರ್ಟೆಂಟ್ ಒನ್. ವಾಟ್ ಆರ್ ದ ಕಾಂಪ್ಲೆಕ್ಸಸ್ ಆರ್ ದೇರ್? 5 complexes are there. Number 1, complex number 1, the NADH dehydrogenase. Complex number 2, FADH2 FADH2 complex. Okay. Then complex number 3, that is cytochrome B1 complex. Then complex number 4, that is cytochrome C1 and other things are there. No, that is cytochrome 5 ಯಾವುದು? ಸರಿ, complex 5 ಯಾವುದು ಅಂದ್ರೆ F0 F1 particles. Okay. So these are the complexes. So these all are the things. Again, one more thing to be explained. ಇಲ್ಲಿ ನಿಮಗೆ ಗೊತ್ತಿರಬೇಕು, ನಾವು ಸಾಮಾನ್ಯವಾಗಿ ಈ ಬ್ಯಾಲೆನ್ಸ್ ಶೀಟ್ ಹೇಳಿದ, ಬ್ಯಾಲೆನ್ಸ್ ಶೀಟ್ ಈಗಾಗಲೇ ನಾನು ಹೇಳಿದೆ. ಏನ್ ಗೊತ್ತಾ, ನಿಮಗೆ ಒಂದು ಕ್ರೆಬ್ ಸೈಕಲ್ ಆಗುವಾಗ ಎಷ್ಟು NADH2 ಬರುತ್ತೆ ಅಂದ್ರೆ 4 NADH2. 4 × 3 12. ಒಂದು ಅಂದ್ರೆ 1 ಪೈರುವಿಕ್ ಆಸಿಡ್ ಇಂದ 1 ಪೈರುವಿಕ್ ಆಸಿಡ್ ಗಿವ್ಸ್ 4 NADH2, 1 FADH2, 1 GTP. ಇನ್ನೊಂದು ಪೈರುವಿಕ್ ಆಸಿಡ್ ಇಂದ 4 NADH2, 1 FADH2 and 1 GTP. Okay. 4 plus 4 8. 8 NADH2, 2 FADH2, 2 GTP. GTP means ATP. Okay. ಇದಿಷ್ಟು ಲೆಕ್ಕ ಮಾಡಿದ್ರೆ 30 ಬರ್ತದೆ. ಕ್ರೆಬ್ ಸೈಕಲ್ ಸರಿ ಯಾವುದು? ಗ್ಲೈಕೋಲೈಸಿಸ್ ಅಂದ್ರೆ 6 ATP ಬರ್ತದೆ. 6 ಅಲ್ಲ, ಆಕ್ಚುಲಿ 8 ಬರ್ತದೆ. ಅದನ್ನ ನೀವು ಫೈನಲ್ ಮಾಡುವಾಗ 2 ಕಳಿಬೇಕು, 36 will come. Okay. That is our balance sheet. Okay. ಇದಿಷ್ಟೆಲ್ಲ ಆದ್ಮೇಲೆ there is one more thing to be explained. ಇದೆಲ್ಲ ನಾನು ಈಗ ನಿಮಗೆ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಿದ ಅದು ಮಾತ್ರ ಇರೋದು, nothing more than that. There is a word amphipholic pathway ಅಂತ ಉಂಟು. What do you mean amphipholic pathway? ಇದು ಎಸ್ಪೆಷಲಿ ರೆಸ್ಪಿರೇಷನ್ catabolic pathway, breaking down of larger molecule into smaller ones. But meanwhile, especially in Krebs cycle, ಆ ಎಲ್ಲ ಜಾಗದಲ್ಲಿ ಆಯ್ತಾ ಏನು catabolic ಆಗುತ್ತಲ್ವಾ, catabolic ಆಗುವಾಗ sometimes some of these molecules are used for the synthesis of fat. Some of the molecules are used to synthesize protein in a reverse order. Synthesis of protein, synthesis of fat is an anabolic process. Okay. ಈಗ ಆಲ್ಫಾ ಗಿಟೋಗ್ಲಿಟರಿಕ್ ಆಸಿಡ್ ಇದೆಯಲ್ಲ, ಅದನ್ನ ತಕೊಂಡು ಮೆನಿ ಆಲ್ಫಾ ಗಿಟೋಗ್ಲಿಟರಿಕ್ ಆಸಿಡ್ ಯೂಸ್ ಟು ಫಾರ್ಮ್ ಪೈರುವಿಕ್ ಆಸಿಡ್, ಸಾರಿ ಅಮೈನ್ ಆಸಿಡ್ಸ್. ಅಮೈನ್ ಆಸಿಡ್ ಫೈನಲಿ ಫಾರ್ಮ್ ದ ಪ್ರೋಟೀನ್. ಹಾಗೆ ಪ್ರೋಟೀನ್ ಸಿಂಥಸಿಸ್ ಆದ್ರೆ ಅದು ಅನಾಬಾಲಿಕ್ ಪಾತ್ವೆ. ಪ್ರೋಟೀನ್ ಬ್ರೇಕ್ ಆದ್ರೆ ಕೆಟಾಬಾಲಿಕ್. ಇಲ್ಲಿ ಅನಾಬಾಲಿಕ್ ಆಗ್ತದೆ, ಕೆಟಾಬಾಲಿಕ್ ಆಗುತ್ತೆ. therefore it is called amphipholic pathway. So ಟೈಮ್ ಕಡಿಮೆ ಇರೋದ್ರಿಂದ ಸ್ವಲ್ಪ ಸ್ಪೀಡ್ ಆಗಿ ಹೋಗ್ತಾ ಇದ್ದೇನೆ, otherwise it could not complete. Okay. Then final things ಅದೇ ಇರೋದ್ರಲ್ಲಿ one more thing is the respiratory quotient. How much carbon dioxide will you release by using certain amount of oxygen? That's what we call RQ value ಅಂತ ಕರಿತೀವಿ. ಆಯ್ತಾ? The ratio of the volume of carbon dioxide evolved to the volume of oxygen consumed. It's called respiratory quotient. RQ value is equal to carbon dioxide bar O2. ಸಾಮಾನ್ಯವಾಗಿ ಕಾರ್ಬೋಹೈಡ್ರೇಟ್ is the substrate molecule. ನಿಮ್ಗೆ ಈಕ್ವೇಶನ್ ಗೊತ್ತಿರಬೇಕು, C6H12O6. ಇಲ್ಲಿ ನೋಡ್ಕೊಳ್ಳಿ, ಎಷ್ಟು ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಇದೆ? ಕಾರ್ಬನ್ ಇದೆ 6 ಕಾರ್ಬನ್. ಸರಿ, ಎಷ್ಟು ಆಕ್ಸಿಜನ್ ಯೂಸ್ ಆಯ್ತು? 6 ಆಕ್ಸಿಜನ್ ಯೂಸ್ ಆಯ್ತು, 6 ಕಾರ್ಬನ್ ರಿಲೀಸ್ ಆಯ್ತು. CO2 ಬಾರ್ O2 ಅಂದ್ರೆ 6 ಬಾರ್ 6 is equal to 1. RQ value for carbohydrate is 1. ಪ್ರೋಟೀನ್ ಫ್ಯಾಟ್ ಆದ್ರೆ ಲೆಸ್ ದೆನ್ 1. ಪ್ರೋಟೀನ್ ಆರ್ ಫ್ಯಾಟ್ ಆದ್ರೆ ಲೆಸ್ ದೆನ್ 1 ಇಲ್ಲಿ ಕೊಟ್ಟಿದ್ದಾನೆ. 0.7 ಉಂಟು. ದಟ್ ಇಸ್ ಅ ಟ್ರೈಪಾಲ್ಮಿಟಿಕ್ ಆಸಿಡ್. That is a fat, that is 7 ಇರೋದು. If it is a protein, it is 0.9. ಅವೆರಡು ಸಹ less than 1. And if it is a substrate molecule, is organic acid ಮತ್ತೊಂದಾದ್ರೆ it will be more than 1. So this much of thing is there. I think if there any student is there to ask a question, Please ask. I will answer. I will try to answer to maximum. Hello. Hello.
ಶಿವರಾಜ್ ಸುತಾರ ಅಂತನವರು ನೋ ಸರ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ ಸರ್ ಆನ್ಸರ್.
Hello, sir.
ಶಿವರಾಜ್ ಸುತಾರ ಅಂತೇಳಿ ವಿದ್ಯಾರ್ಥಿ
ಹಾ.
ನೋ ಸರ್ ಅಂತ ಕೊಟ್ಟಿದ್ದಾರೆ. ಮತ್ತೆ ಇನ್ನೊಬ್ರು ಅನನ್ಯ ನಾಗರಾಜ್ ಅವರು
ಹಾ.
ಸರ್ ವಾಟ್ ಇಫ್ ATP is produced asking question.
It's a phosphorylation. It's oxidative phosphorylation. When the ATP is synthesized during respiration, it is called oxidative phosphorylation because food molecules are oxidized. When energy is released during oxidized and that energy is used for the synthesis of ATP, it is called oxidative phosphorylation. The same thing if happen in the photosynthesis is called photophosphorylation. Any other question, sir?
No sir.
And is there any feedback from the students, sir?
Sir,
Is it,
one more question from Ananya Nagraj.
Yeah.
ATP is not produced. I think she's give answer. You're asking one more question, sir.
ATP?
ATP is not produced.
Then what happened? Is the question. Sir, question is not clear. ATP, if it is not produced, we have no energy to do any work. Even plant or animal, whatever it may be. ATP is energy currency. ATP is ultimate energy source to all activities of both any living organisms. ATP is must.
Okay, sir.
Okay.
ಸೊ ಯಾವ್ದೇ ಕ್ವಶನ್ಸ್ ಇಲ್ಲ ಸರ್. ನಮ್ಮ ಮಕ್ಕಳು ಏನಾದ್ರು ಕ್ವಶನ್ಸ್ ಕಳಿಸಿದ್ರೆ ನಾವು ಮೇಲ್ ಮುಖಾಂತರ ತಮಗೆ ಇಮೇಲ್ ಮುಖಾಂತರ ಕಳಿಸ್ಕೊಡ್ತೀನಿ.
ಆಯ್ತಾಯ್ತು ಸರ್. ಓಕೆ ಸರ್. ಈಗ ಅರ್ಥ ಆಗಿರ್ಬೋದು ಸರ್. ಮಕ್ಕಳೇನಾದ್ರು ಮಕ್ಕಳ ಫೀಡ್ಬ್ಯಾಕ್ ಗುಡ್ ಆರ್ ನಾಟ್?
ಎಸ್ ಸರ್.
ಓಕೆ. ಸೊ ದೆನ್ ವಿಲ್ ವೈಟ್ ಅಪ್ ಸರ್.
ಎಸ್ ಸರ್. ಎಸ್ ಸರ್.
ಮಾಡೋದಾ
ಸರ್? ರಮೇಶ್ ಸರ್. ಹಲೋ. So anyway, thank you, sir.
Okay, thank you, sir. Okay, welcome.
So today I give a very good talk, very good informative talk on today's topic. So once again, I thank myself on behalf of KSTA. Thank you, sir.
Okay, welcome, sir. Thank you.
So I thanks to all the attendees, and I thanks once again our KSTA CEO, sir, Dr. A.M. Ramesh, sir. Thank you, sir.
Bye, sir. Bye.
ವಿದ್ಯಾರ್ಥಿಗಳೇ, ನಾಳಿನ ಉಪನ್ಯಾಸ ಕಾರ್ಯಕ್ರಮದಲ್ಲಿ ಡಾಕ್ಟರ್ ಬಿಜು ಧರ್ಮಪಾಲನ್ ರವರು ಕ್ರಾಫ್ಟಿಂಗ್ ಎ ಕೆರಿಯರ್ ಇನ್ ಸೈನ್ಸ್ ಎಂಬ ವಿಷಯದ ಮೇಲೆ ಉಪನ್ಯಾಸ ನೀಡಲಿದ್ದು, ಸೊ ಹೆಚ್ಚಿನ ಸಂಖ್ಯೆಯಲ್ಲಿ ತಾವೆಲ್ಲರೂ ಹಾಜರಾಗಬೇಕೆಂದು ಮತ್ತೊಮ್ಮೆ ನಮ್ಮ ಅಕಾಡೆಮಿ ವತಿಯಿಂದ ತಿಳಿಸಿದೆ. ಸೊ ಓಕೆ, ಥ್ಯಾಂಕ್ ಯು.